So here, there's more concentration of solutes outside the cell. The water inside the cell is like, wow, we gotta fix that. So it goes out. And the cell shrivels up. If I had the eggs ready, we were going to do this in lab, but unfortunately the eggs did not get going in time, so we're going to have to save that till next week. But you can see here on these red blood cells, this is the normal shape. Here is the shrunken one in the hypertonic solution. Here's the swollen one in the hypotonic solution. Now, the eggs, you wouldn't see an obvious change like that for the amount of time we're going to put them in, so we would weigh them. And if they weighed more, it would mean they had taken in water. If they weigh less, then they've lost water. Here's another set of illustrated cells, and green arrows for water leaving the cell, yellow arrows for water entering the cell.
This membrane separates pure water on the left from a solution of sucrose and water on the right. The membrane is semi-permeable. Water can cross it, but sucrose is too large for the membrane's pores.
Just go ahead.
Water molecules will move from a solution with a lower concentration